In this video, you will learn about three main ways the central nervous system is supported: the meninges, cerebrospinal fluid, and the blood-brain barrier. Together, these structures provide physical protection, chemical stability, and selective control of what enters or leaves. First, the brain is protected by the bones of the cranium, but beneath those bones are three connective tissue membranes known as the meninges. These layers surround both the brain and the spinal cord. The most superficial layer is called the dura mater. The middle layer is called the arachnoid mater, and the deepest layer of meninges is the pia mater. The dura mater is the superficial, tough, fibrous connective tissue layer. In the brain, it has two layers that are usually fused together, except in regions where they separate to form dural venous sinuses. These sinuses act as drainage channels, carrying cerebrospinal fluid and blood out of the brain and into the internal jugular vein. The dura also forms dural septa, which are inward extensions that stabilize and limit movement of the brain. For example, the falx cerebri is a dural septum in the longitudinal fissure that separates the two cerebral hemispheres and anchors to the crista galli of the ethmoid bone. The subdural space deep to the dura mater is a potential space that normally does not have any contents. However, a subdural hematoma occurs when blood fills this space. Trauma to the brain can rupture blood vessels, leading to a subdural hematoma. This puts pressure on the brain and can be a serious, life-threatening condition if the blood is not drained. Deep to the dura is the arachnoid mater, a loose, web-like middle layer. Beneath it lies the subarachnoid space, which contains cerebrospinal fluid and blood vessels. Small projections called arachnoid granulations or villi extend through the dura mater into the dural sinuses. These granulations are essential for reabsorbing CSF back into the venous blood. The pia mater is the deepest meningeal layer. It is thin and delicate, clinging tightly to the surface of the brain and following every gyrus and sulcus. The pia forms a barrier that separates the brain's extracellular fluid from the cerebrospinal fluid, while still allowing the exchange of small solutes like nutrients and wastes. Around the spinal cord, there is an epidural space between the dura mater and the vertebral bones. This space contains adipose tissue that cushions and protects the spinal cord. Epidural anesthesia, commonly administered before childbirth, is an injection of medication into the epidural space to block transmission of sensory information up the spinal cord. Now that we've covered the meninges, let's look at the fluid that circulates between them in the subarachnoid space. This liquid is known as cerebrospinal fluid, commonly abbreviated CSF. CSF cushions the brain and spinal cord, and it helps transport nutrients, gases, and waste products. CSF is produced and contained within the ventricles, which are interconnected cavities lined with ependymal cells. The right and left lateral ventricles are large, C-shaped chambers within the cerebrum. They're separated by a thin membrane called the septum pellucidum. The third ventricle lies along the midline between the medial walls of the thalamus and hypothalamus. The cerebral aqueduct passes through the midbrain, connecting the third and fourth ventricles. The fourth ventricle lies between the brainstem and cerebellum and is continuous with the central canal of the spinal cord. Inside the ventricles, CSF is produced by the choroid plexus, a cluster of capillaries covered by ependymal cells. CSF flows from the lateral ventricles through the interventricular foramina into the third ventricle, then through the cerebral aqueduct into the fourth ventricle. From the fourth ventricle, CSF passes through the median and lateral apertures to enter the subarachnoid space surrounding the brain and spinal cord. Finally, CSF is reabsorbed through arachnoid granulations into the dural sinuses, returning to the bloodstream. This continuous cycle ensures that the brain is constantly bathed in fresh, filtered fluid that maintains pressure, removes waste, and cushions delicate tissues. Lastly, we'll look at the blood-brain barrier, a critical structure that protects the neural environment. The blood-brain barrier is formed by tight junctions between the endothelial cells of brain capillaries. Astrocytes release chemical signals to stimulate the formation of tight junctions between the endothelial cells. These tight junctions prevent free exchange between the bloodstream and the extracellular fluid of the brain, The blood-brain barrier is important to prevent harmful substances and pathogens from entering brain tissue. It blocks large polar molecules and even immune cells like leukocytes. The blood-brain barrier maintains the brain's chemical stability, which is essential for proper neuronal signaling and protection against toxins or infection. The blood-brain barrier permits small nonpolar molecules such as oxygen and carbon dioxide to cross by diffusion. It also allows water-soluble nutrients like glucose and amino acids to cross, but only through specific membrane transporters. To summarize, the meninges protect and support the brain and spinal cord. Cerebrospinal fluid cushions, nourishes, and cleanses the CNS. The blood-brain barrier maintains the brain's internal environment by tightly controlling what crosses from blood to neural tissue. Together, these three systems create a secure environment for the brain and spinal cord.